Okay, now I just need to be able, there we go. Okay, so why focus on topological maps? Well, if you read my bio, you know that I was a geologist originally. So I did mapping, and there's layers, and there's sulci, and there's gyri. But then I actually went into late Phil Ulinsky's lab, and he worked on turtles. And so we have to have just 30 seconds on turtles. And so the very first thing I did was to look at the cholinergic projection from nucleus isthmi up into the tectum. And part of the thing that attracted me to this was being able to draw these beautiful terminals. These are ones that Cahal called terminals like paintbrushes or brooms. And they actually form, they're about 1/10 of a millimeter across, and they form a nice topological map of the retina onto the surface of the tectum. Now, I knew from the beginning there were some non-topological maps in the brain, but I had argued that there are basically no places in the brain without maps. And maps are really the first principal component of brain organization. So I always start off, hat tip to Andrea, got to get into the basal ganglia. So here's a little projection coming from the putamen down into the globus, the two parts of the globus pallidus. There's 4 maps that are formed from single neurons in the putamen. Here's a little injection into the caudate nucleus. There are 2 injections, and you can see if you go down to the substantia nigra pars reticulata, the GABAergic part, you can see that there's a map there. But the dopaminergic, the projection to the dopaminergic substantia nigra is topographic as well. And I always say, if your middle finger did something wrong, why punish your index finger? You need topography. Now here's the cerebellum. We'll talk about that toward the end. And here's some of the structures that project to it. If you look at the bottom one, the inferior olive, and if you look at it in the cross section, you can see it's basically a cortex. It's a 2-dimensional cortex that's been folded up and jammed into the brainstem because it needs to preserve a 2-dimensional map across its surface. Even the olfactory system has maps. So the receptors aren't mapped, but they actually sort themselves into a map when they hit the olfactory bulb. So we'll take a whirlwind tour through maps, all multisensory maps, mouth maps. And then I'll talk a little bit about trying to find maps with quantitative T1 or DTI in the gray matter. And then I'll talk a little bit about sort of cognition and language in maps, and then a peek at the cerebellum and maybe some visual maps in S1 at the end. So the cortex is a thin 2D sheet with overwhelmingly local connectivity. So here's an owl monkey, and this is some work that I did many years ago. If you look at where the star is, that's a blood vessel. And so now we'll look at a section, a myelin-stained section perpendicular to the surface. Looks like that you can see the blood vessel under the star. Some of the areas are relatively easy to find. So you can see, the V1 border is very clear at the arrow. But a lot of other ones are less easy to find. And so Roger Sutel and Jaime Olavarria and myself developed these methods of peeling the cortex off of the white matter and flattening it and then sectioning it parallel to the layers. And if you do that, you can start to see these subtle differences between different areas. You can see the V1 border very clearly. V2 is a little harder to see. If I sort of flash back and forth, you can sort of make it out. And MT is pretty clear. But if you look there's a whole bunch of areas there that are pretty hard to see. And you can only really find those by mapping. And you've got to map at a very high resolution to pick out these areas. And if you do that, what you see is topological re-representations of the retinal surface across the cortex. And some observations about these areas are often very small. So here's an area that's about 1 millimeter across. It's got a whole quadrant representation in it. There it is in a section. So left, right is the map coordinate. And you can see that the actual map coordinate of a whole representation of a quadrant is actually smaller than the thickness of the cortex. And if you look in mice, they have these unbelievably tiny little visual areas that are just like a tiny little tube going down through the cortex that have a whole representation, a systematic representation of the visual field. So some areas you can find with myelination, like MT, that's the black border compared to the sort of purpley mirror image, non-mirror image map of the visual field that you have in MT. But, you know, that's MT, even though it's less myelinated, and that's not MT, even though it's actually myelinated. So you got to do mapping. These areas vary a lot between individuals. So if you look, this is a small area that has an upper field representation in it. Here's another animal, exactly the same size animal. That area is only about a quarter as big in this other animal. But you might be wondering, how do these sort of continuous, fine-grained continuous maps comport with smaller, you know, these little patchy or modular kind of connections that you see in the cortex? And so you've got, you know, blobs in V1 and stripes in V2 and patches in MT. And it turns out if you inject anywhere in the cortex, you could be in S1, M1, prefrontal cortex, you know, V1, you see these little patchy connections, sort of modular connections. But the thing that you kind of miss when you look at this is that over 90% of the connections are made within that 1 millimeter radius circle. This includes all the long distance connections in the cortex too. So the cortex is overwhelmingly locally connected. It just happens that our MRI voxels are about the size of one of those little locally connected patches, and that was kind of handy. But the problem was, as Andrea mentioned, we had to sort of deal with this nasty folded cortical surface. And so in the early days, I just peeled it off. This took about 12 hours. And I painted it so I could see where the gyri were. So even before I did any computer cortical flattening, I knew it could be flattened because I had actually physically flattened it. This is the calcarine. I cut the calcarine apart so that it would lay flat. Then they gave me one of these, which is less smelly. And in the early 90s, Anders and I, mainly Anders, developed these methods of reconstructing the cortical surface. And it was initially for EEG, MEG, not fMRI, because the fMRI didn't exist at the time. But I went on to try to turn the brain into a regular shape. So here we inflate the brain, and then this was my first attempt to turn it into an ellipsoid. I'm not as good of a programmer as Anders, but I eventually got it working. And then once you had it as an ellipsoid, you could look at where the major sulci were before you blew it up, and then use that to actually drive a nonlinear alignment between brains. And this has a big advantage in that you can, there's the alignment process, is that you can align an area that is a sulcus in one brain, but a gyrus in another. That's actually quite hard to do in 3D. Now, Bruce Fischel rewrote these programs much more robustly, and that's why FreeSurfer is used by so many people. One of the things he did was to get rid of the damn ellipsoid and do a sphere. You can do it, it's much easier to deal with a sphere. And then you can apply a coordinate system to it. But another more subtle point is that when you do surface-based mapping, you get rid of all these little crinkles. The cortex is crinkled. You have major sulci, and then there's all these little crinkles, and you can see that back in this flattening here. You can see all those little crinkles in the parieto-occipital sulcus. There's a main sulcus, but then all these little bumps, and that really kind of sort of smears out the data in 3D. But it also has effects on the surface. So this is a surface that some of you might recognize as the FreeSurfer sort of average surface made by surface averaging surface coordinates. But it turns out it's a shrunken head because it got rid of all the crinkles. And so when you blow it up, it only has like about two-thirds of the surface area of a real hemisphere. And so I know I want to use this, but more people should use it. This is inflate everybody and then average the surface coordinates. And then you don't lose that surface area. You don't get a shrunken head. But you know, very soon after we wrote these programs, functional MRI became possible. And so we developed various surface-based pipelines where we sample the data onto an individual subject's surface and then morph it into this spherical coordinate system, and then do all our analysis of our complex valued data in that standard coordinate system, and then view it back on one of the surfaces. First thing we did, of course, was just try to find these early visual areas, which we knew were there from primates. But from the beginning, I really wanted to go to higher visual areas and use a task. So one of the those early scans didn't have any task aside from fixation. So this is a remembered saccade location task where you have to remember where you're going to make a saccade to. And it turns out you see a very clear map in human LIP of remembered saccade location, hemifield map. The physiologists weren't really expecting to see a map. And part of the problem is the receptive fields are relatively big. People don't record a lot of different places. But I have one big advantage is that I have 1 million neurons per voxel. And so I get a nice spatially resolved signal from that, or a sort of a noise-free signal from that. So when this first came out, a lot of neuroimaging people said, there's no way that's LIP. And why is that? It's because LIP is supposed to be the lateral interparietal area from macaque monkeys, whereas in humans, it's on the midline. So why does it get there? Well, there's a complicated problem of mapping between species. So you've got this large, so-called default mode area, and that's expanded in humans, and that kind of pushes LIP toward the midline. From the beginning, we wanted some kind of generic method of using more complex visual stimuli, because a lot of the higher areas don't really care too much about checkerboards. And so we used videos or something like this, where it's a horrible task where you're monitoring for an occasional number and an occasional upside down face, all while looking at that central black point. So if you do that, you get lots of areas spread out across the lateral surface of the cortex. And I'm going to use this kind of red for upper, green for lower for a lot of the next pictures. So if you look at these areas across people, they're not exactly identical. So all humans don't have exactly the same number of areas, but they're close enough so that if you average, this is a 24 subject complex value, polar angle average, you can easily make out all these areas. So there's the original LIP with the upper field sort of anterior, but there's multiple LIPs, and there were multiple VIPs, ventral intraparietal areas. And so let's talk about those for a minute. So these were known to exist from work in the 90s, that there were multisensory areas that represented visual space and somatosensory space in the same neurons. And they're right where you would expect them to be, right at the boundary between visual and somatosensory cortex. There wasn't really the conception from the animal data that there was actually a map in these areas, as opposed to just individual neurons with paired receptive fields. And so Raisong and I made this fine device to air puff the faces of the CogSci graduate students. And sure enough, there's a map there right behind the central sulcus in sort of upper parietal cortex of the face. But if you then do a visual map, that's an air puff map at the top, but there's a visual map, you can see that those maps are very closely aligned with each other. But you might wonder, like, what if your eyes are looking somewhere else? That should misalign the maps. And so the third case there is to actually look at the center of a video while it goes around in a circle, very close to your face. We're talking like 2 inches away from your face. So a visual object is slowly moving around your face, but the retinotopic stimulation is approximately constant. And if you look at the response to that, you can see that all three maps those three radically different maps are very similar to each other. So you can fixate the center, have something go around, do air puffs in the dark, or fixate the center of a video so that it doesn't change its retinal location, but just goes around your face and you see the same thing. And so really what you have here is a face-centered map where you've essentially taken visual maps and jiggered them into alignment with a somatosensory map using eye position information. There's actually probably 4 maps in area VIP. You can see we're getting up quite close to the central sulcus, up to where somatosensory only maps are, although not completely, as we'll see at the end. So if you map these in great detail, you can see they're slightly different between different people. But again, they're close enough so we can average across subjects in the surface-based coordinate system. So if you look at VIP, it's interesting that this was a very ticklish experiment, I can attest. If you look at the map of the face in VIP, it's quite big. If you look at the map of the lips, very small. But if you look at S1, you can see the lips are huge still in S1. And so what's happened is that the somatosensory maps themselves have been sort of adapted to generate a more veridical representation of space than you have in S1. Ray Song went on to make a 64-channel body map device, AirPuff body map device. And it turns out there's legs and calves all the way down to the toes in VIP. So if we look at the little homunculus at the bottom there, you can see you basically have a face, and then you have the shoulders and the axial body, and then the legs and the toes overlaying a visual map that goes all the way down to the very far inferior periphery. But then the movable arm is actually sort of stuck off to the side from the axial body and the face. And again, this is not totally surprising, because we know from experiments in animals that if you stimulate that medial part of parietal cortex, you get hind limb movement. This is actually from an experiment in a bush baby. And if you actually have somebody reach out in the scanner toward an object, you get a massive activation of prefrontal and parietal areas just from a simple reach toward an object. So these areas are obviously involved in shaping actions, not just sensing, sensory things. So always in an effort to go where no one has mapped before, we've done some mapping of the inside of the mouth. This is me saying pa 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 in the scanner. And this is me saying cha 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 in the scanner. You can't see my teeth, so it's a little disturbing without the teeth. So I've added them back in. And so now if you have somebody say words or sounds, syllables in response to an auditory cue that start at the front of the mouth, so pa, fa, fa, ta, cha, cha, ka, you can see that the place of articulation is moving backwards. So there's an auditory cue, and then you go pa, 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 and then the auditory cue says, fa, fa, and then you go fa, 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 fa. So if you look at the auditory cue, which is happening every 9 seconds, you can see a big signal in primary auditory cortex, nothing in the mouth. But if you look at the signal that's periodic at the rate of once every 64 seconds, you can see a map of the mouth. And interestingly, it's not just S1, where you might expect that the palate and the tongue were represented straightforwardly, but you can see it in M1 as well. So there's a very systematic representation of place of articulation in M1, even though the muscular structure of the tongue is quite complex compared to the somatosensory representation of the palate. So we've done this across subjects. So if you look across subjects, these maps, of course, align across subjects. You can actually, there's a lot of work now with ECOG showing that you can actually predict what consonant somebody is articulating by looking at the patterns right here in basically primary motor cortex, primary somatosensory cortex. But when you average across people in the left hemisphere there, you can see you start to get some other areas in the cortex that have some representation of place of articulation. So let's turn to tonotopy. So tonotopy is quite different because auditory maps have a one-dimensional receptor sheet. And so the connections in the brain tend to be point-to-plane connections rather than point-to-line connections like you have in the 2D receptor sheet case of the visual and somatosensory system. So you can just do simple tones, which we did early on, and you can sort of see some of the early auditory areas. But by analogy with the windowed videos, you can do bandpass filtered vocalizations or pop music. So pop music has like an unbelievably flat input spectrum in an attempt to make it as loud as possible. So like that works really well too. And if you do that, you can actually see that these tonotopic maps extend well down into the STS, covering a large patch of cortex, much bigger than primary auditory cortex. We've done attention-atopy analog experiments. So here you have an actual pitch that's changing pitch, but in this case, you're presented with a full spectrum, and you just attend at different times to different frequencies, and so you can see that you see virtually the same maps, and even strongly right in primary auditory cortex to just the effect of attending to a particular frequency. And the auditory system really plays around with maps. So here's an example from an owl where you've got a frequency going this way, but you have characteristic delay, basically sort of spatially mapping out delays. If you look at where that structure is in the auditory system of the owl, if you go a little forward, you see a remarkable place where these point-to-plane projections have been rotated 90 degrees with respect to each other from two different nuclei. So one measures interaural amplitude and the other one measures interaural time delay. And the result is to construct a two-dimensional map, which the nervous system says, well, now I got a 2D map, like a visual map, and I can send it over to the spirit colliculus. So there's a lot of playing around with maps at all different levels. Now, there's a lot of work to be done here. We don't really know it's orthogonal to tonotopy. People have had various suggestions, and we don't know whether there are different kinds of higher level maps like have been seen in bats, in upper auditory areas in bats. And so those are interesting things. We've tried a couple of things. They didn't work. Otherwise, I would have told you about them. Okay, so let's turn over quickly to trying to find these areas with quantitative T1 or diffusion. So the quantitative T1 methods, why do we want to do this? Well, the problem is if you just get an image out of the scanner, it's just a number. And there's variation, particularly in how the RF gets into the head in different parts of the head that can change the effective flip angle. And you can try to fix this afterwards, but it's a big problem. It's like a 25% variation in flip angle because of the way that the RF gets into the head at 3T. So there's several ways of fixing this, but the way we did was to use two different flip angles and then try to essentially solve for the T1 and proton density. So here's the signal that you get out as a function of flip angle, and it's got a nasty looking equation. But working with other mathematicians much better than me, they showed me how this equation can be turned into this equation. It's amazing that those could be the same thing. And get rid of all the exponentials, and then you end up with basically a linear equation. And once you have a linear equation, you can then do two different flip angles and solve for T1. And if you do that, the problem is you still have to deal with that problem of the RF penetrating differently into different parts of the head. So you do yet another scan with two different kinds of echoes that have different equations for them, and then you can basically solve for the flip angle and then stick that back into those first two equations on the previous slide. So if you do that, you can get back to Flechsig 1920, except that he was working on dead people. So now you can actually sort of visualize these early myelinating areas in living humans. And so one of the first things we found was you could pick up the myelination difference in different layers of the cortex. And so if you look at a myelin stain, you can see that the deeper layers of the cortex are more myelinated. And if we slowly ramp our way through the cortex as we get toward the superficial layers, you can see, for instance, that M1 is a lot more myelinated than S1. We know that was the case, beforehand. So this is kind of a sanity check. The problem with this kind of work to keep in mind is that if you go along the x-axis, those are different cortical areas with different amounts of myelination, and they're systematically different from each other. Different T1 values, different amounts of myelination. But unfortunately, the variation as you go between different layers in the cortex is much larger than the difference between different cortical areas. And so you've got to really nail the level at which you're sampling in the cortex very accurately in order to make this work. But if you do that, And you also have to deal with the fact that the layers change in the sulcus and the gyrus. You can see that the deeper layers in the sulcus thin out, whereas in a gyrus, they sort of poof out a little bit. So you can pick that up in this data. You can see the correlation between curvature and T1. But if you look at some retinotopy, the first surprising thing we found is that lateral spot that a lot of people had identified, or that Van Essen had identified just before he published it as MT, is in fact not MT. It's only about half MT. And that's, again, not terribly surprising because a lot of the areas around MT were known to be pretty heavily myelinated. So there's the actual retinotopic map showing it's only about half of that spot is actually MT. We can see area V6, like Flexig, I note, 1920. So this was an area originally described by John Ullman, which has a big representation of the visual periphery. And so it was discovered also similar area in macaques. But in humans, this somewhat stomach churning stimulus, which I hesitate to think of how it comes over the Zoom, is compared to this scrambled one. So it turns out the structured version of the stimulus very strongly drives this area V6. And those spots that you can see in singular cortex, that the anterior spot is the dorsomedial frontal eye fields. And if you look at the retinotopy, you can see it's got a whole representation of the visual hemifield in it. It also has a representation of eccentricity. This was an experiment where I had to sit in the scanner with my head off to the side with a screen about 2 inches from my eyes in order to be able to get all the way out to the periphery. But if you do that, you can see that the area V6 has a substantial representation of the periphery. You don't see the central activation there until pretty late as the wave is going through V1. So there's the visual map, an average visual map, and you can see V6, and you can also see it, of course, in this T1 or myelination map. You can also see an area in front of it that's involved in reaching called V6A. You can see V3A, which is a very motion sensitive area. It's also very heavily myelinated. And you can see the VIPs, also heavily myelinated, also very motion sensitive. You can see these differences at the individual level without normalization. So you can actually look at a single person over time, for instance. That's another attraction of them. If you've got much better quantitative data this way, you can do less computation. So there's the free surfer standard operation where a whole bunch of normalization steps are done. But on the right, you can see just the data with just a hard threshold, no normalization at all, and you actually get a better surface. So you can do, same subject longitudinal cross-center studies. Everybody's terrified to go to a new MRI center. I can attest, trying to attract people back to SDSU MRI center because of problems like this. So I think these techniques have a bright future ahead of them. What about diffusion? So one of the ways that people typically found cortical areas was to look at the subtly different myelination patterns in different areas, different layers, different amounts of fibers between different layers. And so the first thing, and so if you look at diffusion, there's not much anisotropic diffusion in the gray matter compared to the white matter, but there's some, and it's reliable. So you can see it, if you scan again. And so what we did is, one of the problems is the cortex is folded up. So we need to generate features of diffusion patterns relative to the local normal because the local normal changes all over the place. And we can also use some rotationally invariant ones. But if you combine those and then take patches of the cortex that are at all different angles, and then just train supervised networks to recognize these areas, you can sort of recognize on a rescan, but you can also even classify never seen data. So you can see at the top there, you make a classifier for MT. It can tell that, oh, what's ever in front is not MT. We've also done a lot of unsupervised clustering of these diffusion data sets. So this is the human connectome data set where we sample the diffusion weighted scans to individual surface surfaces and then extract features using the standard spherical harmonics, and then do k-means clustering of that. And so this is the T1 data done by a different method, but a T1 data, and you can see M1 and S1 as you'd expect. This is those clusters of gray matter diffusion features referenced to the local normal. And you can see, going back and forth between those, they find a lot of the same things, but there's enough difference. We probably have an extra couple dimensions to work with, and we've done further classification methods using various parcellations to show that we can do a little bit better job of recognizing cortical areas using this diffusion in the gray matter. But you might wonder, does this, really overlap anything real? So years ago, we did these experiments on two-back tasks in dorsolateral prefrontal. So if you do a two-back identity task, pay no attention to where the thing is, but just see if you can recognize that it occurred again, you get a big dorsolateral prefrontal activation. You actually get less in dorsolateral prefrontal if you do a two-back for location, ignoring identity. But if you actually do a special Marty two-back task where you have to recognize a two-back face while staring at the center so that we get a retinotopic signal out of it, you get a retinotopic signal in that dorsolateral prefrontal cortex that's stable across months, and it's completely smack on the middle of that dorsolateral prefrontal cortex spot that gives you all the big activations when you do a two-back ignoring location. So you can't get rid of location. Location's like a really important thing to keep around. If you look back at our 24 subject average, you can see there's 2 visual maps in that dorsolateral prefrontal cortex area, in addition to another couple maps in the frontal eye fields. So what about route finding? Slightly more cognitive. So this is finding our way around the cognitive science building. So we start at the lab late at night, and then we imagine walking to the bathroom. And then when you get to the bathroom, stay there. And then you then go out of the bathroom and walk to point C. Imagine walking to point C, all eyes closed. And you see, a bunch of activations in the expected place. You see retrosplenial cortex, parahippocampal place area, and some sort of posterior cingulate stuff. You get a lot of some parietal stuff. The frontal eye fields are very active. But if you look at that compared to the maps, you can see that most of those things overlay maps. So a lot of these, something like Imagine Navigation is engaging all these retinotopic maps. But that's sort of just navigation. What about sort of language or something higher? So here's some things on the visual word form area. So if you do slightly eccentric reading versus fixation, what you see is a big spot in the infratemporal cortex in the so-called visual word form area. But then if you do Marty reading, where you sort of like slowly go around the clock and look at that same area, which you can see on the right, which is the left hemisphere, underneath the left hemisphere, you can see there's a map in it. Now, interestingly, if you do this kind of mapping where you just have individual letters, you don't see anything there. So that's really sort of good advertisement for why we want to use complex stimuli, because if you don't use a complex stimulus, there's no activation. Compare that with the actual words going around, you see then suddenly the maps are visible. Here's another version that Ben DeHaas, me and Sam Schwartkopf did. So on the left, we're mapping faces just by slowly scanning down through the face. So see, we're at the eyes and we're heading to the nose. So that's kind of a face feature map. And let's compare that on the right to actually just slowly scanning the eyes down in the visual field without changing, so that we wouldn't get the face feature map on the left. And you can see slightly different than the last experiment, but there's quite a lot of similarity between the 2 maps you get in those 2 cases. And so it suggests that there's actually retinotopy, functional retinotopy in the fusiform face area. But what about language comprehension? That's real, real cognition. So for that, we have to do all the maps. So here's retinotopy, tonotopy, and somatomotoratopy, having people move their body. So here's an example from a single subject, a single long-suffering subject whose name you might know. Here's visual maps, and for reference, here's where the major sulci are. So you're looking at sort of a lateral view of a hemisphere. So you can see the central sulcus and the superior temporal sulcus on both those hemispheres. So now let's do a body map, and you can pick up the supplementary motor areas, and you can pick up the VIPs and the S2s, and then we do a map of the inside of the mouth, and then a map of the different auditory areas, and then paste them all together, and now compare them to reading comprehension. So here's a naturalistic reading comprehension task where we are exactly controlling eye movements. So person just reads these meaningful paragraphs with modulated fixation time by word length, and then suddenly it changes to Hindi. But everything's exactly the same length, and you're making the same, very, very close to the same saccades. And if you look at that versus fixation, you get a lot, but let's look at sort of English versus Hindi. So everything else is the same with all the fixation rates and where you're looking in the visual field, and you see a lot of activation more in the left hemisphere. Let's see where that is relative to the maps. Well, a lot of it overlaps the maps. So we actually did this correctly across about 20 subjects. So here's visual maps, including the dorsolateral, the dorsomedial frontal eye fields in the cingulate sulcus there. Here's our auditory maps, and that includes what I would call the dorsomedial frontal ear fields. Here's body maps with a lot of motion artifact, which we can get rid of with ICA. And if we put the dorsomedial frontal eye fields and the dorsomedial frontal quote-unquote ear fields, you can see they're kind of like on either side of the supplementary motor areas. There's two of them. Then we do the reading task again. And so here's the English versus Hindi. And what do you get? Well, you get the usual suspects. You get some parietal cortex, some dorsolateral prefrontal cortex, some superior temporal sulcus, some infratemporal cortex, some dorsomedial frontal ear field cortex in the cingulate. Now, if you look at where that's related, where that sits relative to the maps, you can see there's a huge amount of overlap with the maps. So you got purple for visual, blue for auditory, and green for somatosensory. But the question is, what if you compare that to something that is a similar kind of serial assembly task? And so here's a case where you're looking at a wordless picture story and having your saccade directed around by a little Gaussian bubble compared to either a scrambled up story or a completely scrambled up picture story. So here's sort of picture versus sort of jigsawed picture. Again, very similar saccades in both cases. And if we take that data and superimpose it on top of the language data, you can see, again, a lot of overlap with maps, but also a lot of overlap with the actual language comprehension activation. And if you actually sort of zoom in on the STS, you can see an interesting contrast. So on the left hemisphere, you see more language-related stuff in the STS, less scene assembly-related stuff, and you see the opposite in the right hemisphere, more scene assembly-related stuff in language. Same way in frontal cortex. Dorsolateral prefrontal looks more languagey on the left, and it looks more sceney on the right. And that's a good thing, because you want to be able to walk and talk at the same time. So you want to be able to have somebody tell something to you, and you assemble a fictive scene in your head while at the same time assembling the real scene that you're walking through without getting too confused. So is there overlap with a lot of these maps? Yes, these maps are heavily involved in these high-level comprehension tasks. So now in the last couple minutes, let's take a quick peek at the cerebellum. So 3/4 of your neurons are there. And if you look at the cerebellum, it looks pretty nice. But if you look at it close up, if you compare it to the cortex, there's the gray matter of the cortex. We've got a couple of voxels to work with at 1 by 1 by 1 millimeter. But in the cerebellum, you're only picking up the major lobules, not the fine folia. So we got a dead one, and we scanned it for about 12 hours. So there's me. That's what the cerebellum looks alive. And that's what it really looks like if you scan with much smaller voxels for a long time. So this is a T2 star weighted representation of the cerebellum. You can see the dentate nucleus in the middle there in the black. That's one of the output structures. Here's a proton density weighted scan that we can use for normalization. And if we combine those and then do some special sauce where we do Anders' famous anisotropic filter in the plane of least variance, then we get this amazing structure. This is the unbelievable surface, folded surface of the cerebellum. And here's going from left to right. That was front to back to front. So here we're in one hemisphere and we're coming up on the vermis. You can see the vermis right about there. And now we're back to the other hemisphere. So it's this amazing spatial map structure. So once we got the initial surface, which I always like to brag, has about as many polygons as the space station in the movie Gravity, is a really irritating thing to deal with, all those polygons. But then we can make a good estimate of the peel surface from the proton density weighted scan, and that's what you get. So this is the first folia level reconstruction of the complete surface of the human cerebellum. Those little creases that you can see are blood vessels. We're just barely small enough voxels. These are 1/5 of a millimeter on the side voxels. And just to put it in perspective, that's about the size of a Purkinje cell, which is perpendicular to those folia that the parallel fibers are running along. So we had to unfold it, of course. So let's blow it up. So it turned out to be extremely hard to unfold. You can see the lobules unfold, but once you actually look at the unfolded surface, it doesn't completely unfold because there's a lot of intrinsic curvature in it. Here you can see those things on the dangling in the middle. Those are called the tonsils of the cerebellum. So yeah, it's extremely hard to unfold. So that now as it goes back down, then you can see all the individual folia again. So it's this very complicated surface. So what are we going to do with that? So I got the little inset from Gravity there. So if you look at this surface, we needed to really flatten it. And so I took out my free surfer chainsaw, and I chainsawed through the surface and got rid of all the parts that weren't cortex and then flattened them. And so here we flattened the anterior lobe you can see at the upper left, and now we're flattening we passed the primary fissure about here, and now we flatten the posterior lobe. But then we get down into the cognitive cerebellum. This is the part that's really expanded in humans, and you can see that's absolutely enormous. And then at the very end, you can see the little tonsils that we flattened out. So this is what it looks like. It's about 1 meter long after you stretch the whole thing out. And it has almost 80% of the surface area of the entire neocortex. Now here's a standard cerebellar atlas at the same scale. It's actually just the white matter, and you can see it severely underestimates the expansion of the cognitive cerebellum in Crus 1 and 2 and lobule 7B and lobule 8. So what's in there, the giant surface area? Well, these are studies done in the 70s and 80s that most people have hardly heard of mapping cerebellum. And this is a single folium, and I'll rotate it sort of vertically. What do you have in there? You have this weird little fractured somatotopic maps. So what does that mean? Well, here's a little 1 millimeter square. And if you look at what that's mapping, it's mapping a small part of the upper lip of a rat, the furry buccal pad. And if you actually look across that 1 millimeter, you can see there is a very clear somatotopic representation of this little patch of skin. And it'll be sitting next to some somatotopic representation of like a hand or a little patch of skin on the stomach. So what are we talking about in the human cerebellum? It's something like that. And so this is a representation of the approximate size of the expected size of these patches. It's just like some absolutely insane hash table in which you take little patches of different parts of the body and sort of patch them all together and then have parallel fibers kind of relating between them. And if you look at the cerebellum in human evolution, you can see that in a macaque, the cerebellum is only about 33% of the surface area of the macaque neocortex, but in the human, it's about 78%. So the cerebellum surface area has expanded much more rapidly relative to the neocortex in human evolution. And it's very likely that that gigantic expansion in the cognitive cerebellum is probably related to, as I said in the paper, in the fullness of time, mentally rearranging equations as a little sort of fictive motor movement to sort of help your higher cognitive things out. I like to say the cerebellum is only 2 synapses away from consciousness. You got to come to the pontine nuclei and then in the cerebellum. So it gets input from a lot of the cortex. Doesn't get input from infratemporal cortex, but it gets input from almost all other parts of the cortex, including orbitofrontal cortex. So it's involved in emotion. And so last but not least, in the last 2 minutes, let's talk about an insane visually driven map in S1. So Esther Kuhn came to me in London wanting to do this experiment. I said, it's not going to work, but it involves a map, so I'm willing to try. And so the experiment was actually touch somebody in the dark with a little piece of sandpaper there, or have a person just view a first-person movie or maybe a third-person movie of somebody else getting touched. And we had to sort of, I had to up my game. So we've got 2 complex valued maps that we have to correlate on the surface, circular correlate on the surface, and we need these geodesically defined spotlights to sort of go over the surface to do the cross correlation in little patches. And we don't want to sort of like cheat by using vertices more than once. So here's a surface intersected with some voxels. There's the actual surface patch. We got to figure out the individual vertices that are representative vertices for each voxel, and then run our little geodesic patch around each vertex in order to calculate these cross correlations. But if you do that, what you see is that if you do actual touch, and then you do watching somebody get touched, you basically only see, if you look at the upper right, you only see the correlation between these 2 maps just in that one little spot, right in S1, right in S1. It's just stunning. It's actually in area 3B and area 1. This is 7T data, so that's why we could see these small maps. Now, the signal is not very big, so if you look at the blue, that's the actual periodic signal from touching digit 2 every 64 seconds. So the signal from seeing somebody's digit 2 being touched is much smaller, as it should be, because it's only about 1/10 as big as the actual signal. And it's in a different layer. The laminar profile is a little different. Now, this was absolute hell to get published. It went through like about 12 or 13 reviews. We had to do a whole bunch of extra things, but reviewer beta side, among friends, there are mirror neurons in primary somatosensory cortex. So what this means is one column contains information from multiple levels of processing. So it's not a good idea to sort of think of like individual cortical areas as being at a certain level of processing. They've got information from all different levels mixed in. It's quite a non-trivial map, especially in the third person view, to somehow get that visual information in V1 and percolate it all the way back down to S1 and get it into the right finger. It's an amazing thing. So in conclusion, topological maps in cognition. So the medieval cognitive neuroscience was wrong. There isn't a common sense. And then all cognition after that, there's just tons of maps, and there's maps that interact with each other, but there's a lot of maps in there. Having a map does not equal unimportant for cognition. And something related to that, think about your visual experience. It's very sharp. It's like V1. It's not like infratemporal cortex. It's not like the very large receptive fields in infratemporal and parietal cortex that are looking out at big chunks of the world. It looks like V1. It's very sharp. And the suggestion is that cognition may penetrate back down to these very early maps, as evidenced by some of this visual map in S1. And in contrast to a well-known psychologist and a computer programmer with lots of Y's in his name who said that that doesn't happen. So this is work involving many, many people over a number of years. And I'll just leave you with a picture of the spherical, extremely distorted spherical cerebellum rotating around. So thanks for that. I'll take questions now.